Dear all, welcome to this video. Please hit the subscribe button so that you automatically get updates for upcoming videos and lectures. Thank you. Enjoy watching.
First, the word bacteriology, it is the branch. Logos, as you all know, stands for studies. So bacteriology, it is a branch and specialty of biology that studies the morphology, ecology, genetics and biochemistry of bacteria, as well as many other aspects related to them. So morphology, obviously the structure of the bacteria. Ecology is the presence of bacteria, that is what is the natural habitat of that particular bacteria. Genetics, whether it is DNA containing bacteria, RNA containing bacteria, what is its genetic sequence, and the biochemistry. Biochemistry here, it means what all nutrients they require after nutrition, what all metabolism they do, what all reactions. Some are nitrogen fixing bacteria, some are aerobic bacteria, so how they work out. So all these things are studied in the term bacteriology. So now what is this bacteria? First of all, a bacteria is a prokaryotic single cell organism. Single cell, as you all know, unicellular, contains only one cell. And prokaryotic, it means it does not have a defined nucleus. I hope everyone knows the difference between prokaryotic and eukaryotic. Prokaryotic are the organisms which have the genetic material, but they don't have defined nucleus. And in case of eukaryotic, they have the well-defined nucleus. They are small in size. Size ranges in around 0.5 to 5 microns. And as you all know, prokaryotic, it means no nuclear membranes, but it may contain DNA and RNA. And they are viewed under microscope. So now what is the natural habitat? As also bacteria are present everywhere in earth, in air, in soil, in water, in hot springs. So everywhere bacteria are present, and bacterial cells are around 10 times smaller than eukaryotic cells, and they are typically, as I told you, 0.5 to 5 micrometer in diameter, and there are around 40 million bacterial cells in 1 gram of soil. So the size being smaller, large number of bacteria can grow in small space. So 1 gram of soil may contain 40 million bacterial cells, and around 1 ml of water, it may contain 1 million cells. So now this is the outline of the structure of bacteria, how it is. So this here, the outer membrane or outer structure is called as capsule. It is just an envelope, but all bacteria do not have capsules. Some have, some don't have. And all those bacteria which have capsule, it is an additional protective covering. Then next is the cell wall. Around this bacterial cell wall usually is present in all the bacteria, but capsule is optional. Then after the cell wall, there is a plasma membrane which bounds the cytoplasmic membrane. Plasma membranes are almost one and the same, which covers the cytoplasm. And here you can see this is the cytoplasm and this is the genetic material. Okay, and these small projections, they are known as pillars or pili. We'll be studying in detail what is the function of them. And this long one is flagella. So again, this pili and flagella, they are optional. It is not compulsory that all bacteria contain pili and flagella. It may not contain also, depending upon the type of bacteria. So now structure of bacteria. So first capsule. So that is a protective, as I told you, it is a protective covering which is made up of polysaccharides. And all those bacteria or microorganisms who have capsules or envelope. In virus it is called as envelope, in bacteria it is called as capsule. So all those microorganisms which have these kind of extra or additional covering, they cannot undergo phagocytosis. So thus the virulence factor is quite high. Then mordants are used in order to stain the capsule, so you can see them under microscope, specifically only capsule. And these capsule materials are antigenic, so they contain antigens. You must have heard about the term surface antigens. So these surface antigens will obviously be present on the capsule surface. Next is the cell envelope. Now this cell envelope is made up of 3 layers, that is the interior cytoplasmic membrane, the cell wall and the outer capsule. So as I told you, cytoplasmic membrane and cell wall will be present in all the bacteria, but capsule will be optional. Now this cell wall is again made up of polysaccharide, and this cell wall defines the shape of that particular bacteria, and it surrounds the cytoplasmic membrane. Does not stain easily by staining techniques, and it does not stain easily, as I told you, and the main function is protection. Next is the cytoplasm. Cytoplasm, you can call it as a reservoir of nutrients. So all the nutrients whatever are required, they will be present in the cytoplasm, and from the cytoplasm all other organelles will be present. They will take whatever they require, and they will also secrete it into the cytoplasm. So it is the place where the cell growth, metabolism and replication will take place. Next is the cytoplasmic membrane. The membrane which covers the cytoplasm. It is made up of combination of phospholipids and proteins, and it regulates the flow of materials in and out of the cell. So if a bacteria wants something as a nutrition, so it will pass through the cytoplasmic membrane and then it will enter into the cytoplasm. So if we take a section of this bacteria, so this will appear. So here again same thing, capsule is present, cell wall is present. Here this is the DNA material, this is the basal body. Here these are the two flagella, ribosomes will be present, cytoplasmic inclusions will be there, pili, these are the pili. Okay, and some may contain or may not contain the folding. It is not compulsory. Next is flagella. So flagella, here you can see the long ones are the flagella, the smaller ones are the pillas. So what is this flagella? This is a hair-like structure which is required for locomotion. Locomotion is movement. So if a bacteria has to move from one place to another, it will move with the help of flagella. So that flagella beats in a propeller-like motion, and it moves towards the nutrients and away from the toxic chemicals. Now what is in this flagella made up of? It is made up of flagellin protein or keratin or myosin. So it is actually a protein material, and the motility can be observed with the help of hanging drop method. Now, what is this hanging drop method? What you have to do is you have to take a slide. On this slide, you will put a drop of inoculum, whatever it may be there, and on the cover slip, what you will do is you will put the jelly, petroleum jelly, any jelly kind of thing, on the four corners of that cover slip, and you will slowly place the cover slip on the drop in such a way that the drop is not dispersed. The drops will remain intact, but you will just cover that drop. Okay, and after that, so that's why that method is called as hanging drop, because the drop is not dispersed. It is kept in its intact state, and then you will see it under microscope. So you can see the motility or the moving microbes in that. Depending upon, it may be axial fibrils. These are the names given to flagella depending upon what state. If its origin is from periplasm, then it is called as periplasmic flagella. If its origin is from cytoplasmic membrane, then it is known as endoflagella. Then axial fibrils, if it is present in the axial position, then it is known as axial fibrils. So depending upon the position, the name varies. Gliding motility is also one of the terms which relates to movement of the bacteria. And taxis is also stands for movement. Phototaxis is movement towards the light. Aerotaxis is movement towards the oxygen. Flagella, now basic structure if we go, then this flagella is made up of three components: filament, hook and basal body. Okay, now as we will be discussing this later, the gram-negative, gram-positive bacteria. But a gram-negative bacteria contains four rings: L, P, S and M ring. L is the lipopolysaccharide layer of outer membrane. P ring is the peptidoglycan layer. S ring is just above the cytoplasmic membrane, and M ring is within the cytoplasmic membrane. Whereas gram-positive contains only S and M ring, L and P rings are missing in gram-positive bacteria. Next is nucleoid. Now nucleoid, as you all know, there is no defined nucleus because this is a prokaryotic bacteria. So the region where DNA is present, that is only known as nucleoid region. And most bacteria have single circular chromosome that is responsible for replication. Next is pili. Pili is plural, singular of pili is pilus. So it is hair-like projections emerging around the surface all throughout, and the outgrowths assist the bacteria in attaching to other cells. So the main function of pili is not movement but attachment. So during replication process, if the two bacteria have to attach, they will attach with the help of pili. Now what is the difference between flagella and pili? So flagella obviously larger in size, pili is smaller in size. These are thicker, these are thinner. These are not attached to the cell wall, these are attached to the cell wall. Flagella origin from the cytoplasmic membrane, pili origin from cell wall. Organ of locomotion is flagella, organ of adhesion is pili. Not required for conjugation. Conjugation is a process of replication. So obviously in pili it is required. Flagella is not required. So, this was the basic introduction of bacteria, the components of bacteria.